All right. Hello, everybody. Welcome to the BRC Workshop Hour. This is our second ever session, so it means a lot that you're here. So throughout this hour, two BRC members will be presenting their workshops in the neuroscience of addiction and technology and the neuroscience of learning. Each presentation will be about 30 minutes, and both will provide time for you all to ask questions and do some fun activities. And also, as you could probably tell, these presentations are being recorded. So if you want to see this content again, there will be an opportunity for you to do that. Without further ado, I'll now introduce our first presenter. All right. So Suman is a clinical psychology master's student from India. She enjoys learning about why we do what we do. So for her, that encompasses cognitive neuroscience and psychology at the moment. She has been in BRC for almost 2 months now, which is a department of SN and called Brain Resource Creation. And she's enthusiastic about being able to create resources to help others learn some of what she's fascinated about. All right, so now I'll hand it over to Suman and we can begin learning about the neuroscience of addiction. Take it away.
Thank you, Erin. Right. So today we're going to look into addictions and what they do to our brain. Not just addiction to substances, but also behavioral addictions. And smartphone addiction, the crux of my presentation is one of the latter. It's a behavioral addiction. So this presentation will be the answer to why are you on your damn phone all the time? Right. So this is a story about a very old story of survival. Back in the days when we humans were quite at a disadvantage when it came to surviving in the wild. Compared to the beasts, we neither had speed nor the power, yet we had to survive. So our brain was the only miracle that kept us alive. Now, this is how a situation would have panned out back in those days. You are a human toughing it out on a hot summer day. There's barely been anything for you to eat these last few days, and you are out of your prehistoric wits looking for some food. So as you wander into the forest, you come across this marvelous smelling, bright red slice of cheesecake. I mean, there's no harm in supplying this imaginary early human with some cheesecake. So you obviously go and eat it. Your taste buds are thanking you, but more importantly, your life has been saved. If not for this sugar and fat-rich slice of food, you would have passed out and probably died. Your brain does all the important work to remember this incident. The place, the taste, the wonderful sense of relief when you had the food. This is so that next time you increase your chance of finding this food again and living a little longer. All of this was about survival. So we'll go on to learn about the elements involved in the process, in this process in our brains. Now you might be wondering how this is related to addictions. So this is a not so old story of addiction as well. So you are not so vulnerable as that human today because you're more likely to find yourselves with a refrigerator filled with food than hunting around for some. This same system that aided your survival has now turned against you and granted you with the gift of addictions. The motivation to eat the cheesecake and the amazing sensation after it, it's still the same. But here, the problem to be tackled, it's not survival, but it's obesity. Okay. Right. How do you know that you're addicted? You can take two things in your mind when I'm explaining this. You can think about a substance, for example, alcohol, or you can think about your smartphone. The former is a substance and the latter is a behavioral addiction. So how do you know you're addicted? The DSM-5, it's the Diagnostic and Statistical Manual of Mental Disorders, which means it lists mental disorders. And it says these are the symptoms of addiction. Well, technically, these are the symptoms for substance use disorders. But this is how it goes. You neglect major rules to use. For example, you are very much into gaming and you neglect your role as a student because you spend way too much time on gaming. repeated attempts to control use are quitting. You keep trying again and again. You don't want to spend hours and hours on the gaming, but you can't. And you keep craving it. For example, you're working on an assignment and you think about, oh, how cool that game was. You want to keep going back to it. Then there's activities given up to use. For example, if you loved playing football and going outside playing with your friends, you wouldn't do it because your mind is occupied with that substance or that behavior. It's there on your mind all the time. Hazardous use, it means that you know it's terrible for you, probably for the people around you, but you still keep doing it. And a lot of time is spent doing this. I remember there was this game called Clash of Clans, and I used to wake up in the middle of the night just to check whether someone has attacked my kingdom, what's going on. So these are a few symptoms, right? Now we are going to find out what goes up in our brains when we are addicted. This is the model we'll use to explain addiction. I'll go slow here. There are three main players in the game. The first is the dorsolateral prefrontal cortex. Dorsolateral. Dorsal means the superior part. There's dorsal and there's ventral. So the superior part of your brain. Lateral is related to the sides of your brain. So dorsolateral. Prefrontal cortex is the region that is in front of your frontal lobe, so somewhere around here. That part of your brain is responsible for making decisions. It's the steering wheel of your brain. For example, it'll say things like, don't do it, that's dumb. You should choose that one. That's something the dorsolateral prefrontal cortex is likely to say. Now, second is the ventral striatum. Ventral striatum, specifically nucleus accumbens, you can see it on the slide towards the right corner. It is the motivational engine. It gets you to pursue goals, generates attraction towards goals. If you have a brownie and you love it, you are going to want it again. So it might say something like, oh, that's amazing. I definitely want it again. And the last player is the ventral tegmental area, VTA. The ventral tegmental area, it's in the midbrain. And this area here is our dopamine supplier. It supplies dopamine. And in a minute, I'll tell you what dopamine does and how it's useful in the system. So three parts: dorsolateral prefrontal cortex, the steering wheel. Second, the ventral striatum or the nucleus accumbens. That's the motivational engine. And the third one is the ventral tegmental area that supplies dopamine. Right. Dopamine is kind of the star of the system. You might have heard that dopamine is the pleasure chemical, but it's sort of a distortion of what dopamine actually does. This is the chemical that, if you remember the cheesecake story from the beginning, it motivates you, directs you, and rewards goal-directed behavior. It is such an all-rounder that if something feels good, for example, eating the cake or hugging another person, it makes sure you learn it pretty quickly. And it might say something like, that was amazing, now let's do it again. So it's quite important when it comes to survival. Right. Zooming into the image. And I don't think I can zoom into the image. I'll just explain maybe if that helps. So just to recapitulate, three parts. And next I will start an activity. Okay. So this activity, who said what? You have to match the part of the brain that is most likely to say the given dialogue. And you can put your answers in the chat. Take about 2 minutes and let's see how you guys do. Okay, go.
Oh no. It looks like the internet's gods were not serving her well today. Let me message her and we'll get started as soon as she gets back. I'm not sure if you all heard the question. I can actually go ahead and present the slides, and so you all we can keep on moving. Let's see. All right. So here's the question. So it's essentially a matching game. So the first one says, the big one is a bad idea. Small is what you should be looking for. Is that A, B, or C? The second one is, here's dopamine, here's some more, and here's a little more. Or those brownies are good, I want more. So it's essentially a matching game between the ventral tegmental area, the striatum, and the DLPFC, or the dorsolateral prefrontal cortex is what that stands for. Put your guesses in chat, and I will try to get Suman, our wonderful presenter, back on the call. All right, so we do have a guess. We have someone saying that number one, oh, we have two guesses now. Oh, awesome, three. And some of them are being sent to me privately. So that's why there's three and not just the one in chat. So we have 1C, 2B, and 3A as one guess, 1C, 2A, 3B as one guess, and then 1C, 2 for the midbrain, and 3 for the striatum. So 1C, 2A, and 3B. All right, let's move on to the answer. Here's why Google Slides are acting up. Oh, looks like we have someone coming in, but it's not Suman. Oh no. All right. We don't actually have let's see. All right. I believe the answer was, let me represent this. I believe the answer was 1C, 2A, and 3B. So congrats to those who got that correctly. All right, let's see if I can move these. Google Slides is so touch and go. All right. I can continue the presentation for Suman. It looks like her internet just cut out completely, so I will pick up where she left off. And I apologize for any interesting words. I don't know this presentation as well as she does, but we have another person, but it's not Suman. All right, so can you all, let's see, get my bearings here. Let's see, just 1 second. My apologies. I'll present. We'll get started in just a few. Oh, she's back. Oh, thank goodness. Okay, my apologies. We'll get started in just another minute. And then she can also explain the quiz. So she's much more familiar with it than I. All right, Suman, welcome back. Are you able to?
I'm so sorry
re-give you
about that.
co-host.
That was unfortunate.
It is
Thank you,
okay.
Ritija.
We're glad you're back.
Okay. Okay. So should I start sharing again? I'll just.
Yes, and we left off on explaining the quiz answers.
Yes. So is this okay? You can you see the quiz thing right now?
Yes.
Okay, cool. So if you have explained the answers, I'll move on to the next slide, or should I do some more work here?
I have not. Go ahead and explain.
Right. So I hope you guys have put in some answers, and I hope you've got them right. So the first one, it says the big one is a bad idea. Small is what you should be looking for. That is something a decision maker would say. And that part, if you remember, is the dorsolateral prefrontal cortex. So in a situation where addictions are involved, your dorsolateral prefrontal cortex might say something like, you probably should stop using your phone and start doing your assignment. Something like that. The second one is, here's dopamine, here's some more, and here's a little more. That's something your dopamine supplier would say. So if you remember, that's the ventral tegmental area. That's a part of the midbrain. So that's C and then A. And then last one, those brownies are so good and I want more. That's striatum. It's the part who loves, who gets more attracted to stuff and wants to follow that. So that was the activity. Now we'll continue. Okay, what is happening between all these parts? Right. So the VTA, the ventral tegmental area, which is our dopamine supplier, it projects dopamine towards the striatum to get it to focus on the goal, block out all the irrelevant information, and narrow down the path to the goal. In addiction, the addictive behavior or substance causes a lot of dopamine to be released, like a flood that is too much for the brain to handle. So normally dopamine is involved so that you learn things quicker. But when you put in an addictive substance or behavior, there's a thing that called dopamine flooding that happens. Brain is flooded with dopamine. Here, the dopamine released by the VTA, the ventral tegmental area, actually reaches multiple parts of the brain. First one is the amygdala. Amygdala deals with emotions like fear, pleasure, anger. The second one that I told you about, the nucleus accumbens, which is a part of the striatum, it's responsible for seeking out a reward. Then we have prefrontal cortex. So dopamine is getting all these parts to do all this stuff. So it makes the prefrontal cortex to make decisions or control elicitation. For example, it will make sure that you have control over the task you're doing. And the last one is hippocampus, which is responsible for formation of memories. So it's as if clicking a brain snapshot of during a pleasurable experience so that it can be gone back to and repeated. The most important thing that dopamine does is long-term potentiation. Long-term potentiation is the word. In simple words, it strengthens the synapses it repeatedly passes through. They are now firm, sensitive, and lodged in place. In daily life as well, if you keep repeating something, you get practice over it. And it's easier and easier for you to do. Something similar happens when you keep repeating the addictive behavior or keep using an addictive substance. The synapses or connections in your brain related to that behavior or substance use get stronger and stronger and lodged in place because of dopamine. So now here's the tricky part. Repeated dopamine flooding will make the whole system hypersensitive to even a slight nudge, like a little cue that reminds you of rewarding behavior. And this will activate the whole system, causing a flood of memory, emotion, craving related to it. Think of it this way. I've experienced, so I loved watching a particular TV show while I was eating my food. So it was the sight of the TV show that made me happy initially. But later on, just the sight of food reminded me of the TV show, and I'd be like, oh, now I'm gonna get to watch the TV show. So dopamine does make sure that different cues associated with the actual rewarding behavior make you feel happy instead of the behavior itself. Right. So because of multiple instances of flooding, the brain adapts to flooding. And it adapts to it by either reducing the number of dopamine receptors or reducing dopamine production. Because our body always wants homeostasis, a sense of balance, not too much, not too less in everything, every system. That's very sad because that's exactly what makes previously pleasurable experiences not so pleasurable anymore. You probably don't enjoy playing outside or reading under a tree anymore. Only the substance or behavior you're addicted to runs the show and causes pleasure. So after I finish a slide, I'll check the chat and I'll address the questions, okay? If they are related to the slide. So what happens when there's repeated dopamine flooding is you lose interest in your other pleasurable activities. You'll stop going outside, stop reading a book. It's sad. So in presence of cues, the prefrontal cortex also does a terrible job at making decisions. The guy who used to make decisions for you, bad at it. This is because of the changes in dopamine production that we discussed about earlier. For example, an alcoholic is walking down a street and he spots a bar. He knows how terrible that alcohol is for him, and yet he will go and get it. Why? It's because of the reduced ability to resist strong urges when he sees the cues. Right. So that was my slide. This slide, I just saw a question in the chat. So in long-term potentiation, is long-term potentiation involved in memory strengthening? Does trauma cause LTP? LTP is, memory strengthening and also behavioral strengthening. If I get better at tying my shoelaces, if I keep doing it again and again and again, there's long-term potentiation in the synapses involved in shoelace tying, because dopamine is released when you learn something as well. And the second part of the question, behavior as well as memory sometimes. Does trauma cause LTP? I'm not sure about it. Maybe we'll think about it more towards the end of the presentation. Okay. Right. I have another activity, like a small activity for you guys. You just have to fill in the blanks and you know what to do. Put your answers in the chat. Go. Hey, good, Nagi Khan, if I'm pronouncing your name correct. Good, you're right. If everyone has answered, I'll also Kalinda, if that's how your name's pronounced, and Rituja, correct. Right. I'll talk about the answers. I see that a lot of you have gotten question. Okay. Yes, Sachin. I see that a lot of you have gotten the second answer wrong. Yes. So the second question says, repeated dopamine flooding increases your ability to experience pleasure in everyday life. Yes or no? The answer is no. Um, it's sort of like your reward system goes haywire, and it's so used to getting so much of it that a normal amount isn't accepted by the system. So you lose the ability to feel pleasure in the little things. You don't enjoy doing little fun things anymore, and that's very sad. And you might have experienced it in your daily behavior as well. If you're addicted to gaming, you wouldn't find slower and subtler activities fun anymore because gaming is on your mind all the time. Okay, so good that you guys got 1 and 3 correct, and a lot of you also got 2 correct. Right, let's move on. Right. So this is a study on addiction and gray matter changes. The picture it might not make sense to you right now, but I'll start talking. It will make sense afterwards. So this was a study conducted by Connolly and others on individuals who had a cocaine dependence. Cocaine is a substance that might cause addiction. So what is to be noted is a reduction in gray matter volume in the right anterior cingulate as the duration of addiction increased. So gray matter, the thing that decreased, gray matter is associated with neuron cell bodies. So those decreased. In the right anterior cingulate, that region of the brain is relevant to us as it deals in affect regulation. Affect regulation is basically the ability to control and manage uncomfortable emotions. Like for example, you can feel very guilty sometimes about something in the past. It's your right anterior cingulate or anterior cingulate that is responsible for you to handle those emotions well. Now, avoidance of those emotions can be quite a motivating factor in developing addictions because nobody wants to go through that discomfort causing feeling. If you successfully somehow avoid that bad feeling, you are going to take that opportunity because that's negative reinforcement, the good feeling that comes out of escaping a terrible situation. So according to the study, the reduction of gray matter volume in this area hints towards a faulty affect regulation mechanism as a result of repeated addictive behavior, just cocaine consumption. Now you can't cope with your bad emotions well anymore. Thus, you keep turning towards the addictive substance. There is a cycle that is established that I'll talk to you in the later slides. Now, this was the left part of the picture in the slide. What is very interesting is how within a few weeks of abstinence, there was an increase in gray matter that was observed in a very similar area, almost as a means of brain's compensation for functional loss. This area is related to self-regulation. So you can see the graph goes up and there was a gray matter volume increase in left cingulate gyrus, very closely related. And it's responsible for self-regulation, which means you might, if you see something related to cocaine, you might have a better chance at stopping yourself from using it. So that's very cool, how your brain copes with situations. Okay. Sure, since we have talked a lot about substances and behavior side by side, I think it's important to address how substance addictions and behavioral addictions do have an overlap, and it makes sense to talk about addictions as a whole and not separate substances and behaviors. Okay, so studies have shown that the major brain changes, cycles are pervasive and form common domains in non-drug addictions. And non-drug addictions are like pathological gambling, binge eating, compulsive buying, internet addiction. All of these are behavioral addictions. And you can find overlaps between these and substance addictions. So what happens is, with time, there is a shift of activation from the ventral striatum to the dorsal striatum. I'll remind you guys, ventral striatum was responsible for, it's the motivational engine. It helps you to pursue goals. So there's a shift of activation from the ventral striatum to the dorsal striatum, ventral to dorsal. So VTA producing dopamine, now instead of projecting dopamine towards the ventral striatum, it projects towards the dorsal striatum. What does this indicate? This marks the change of behavior from impulsive to compulsive. Key terms are impulsive and compulsive. So the difference is, behavior is compulsive when you have the urge to do it repeatedly until a feeling of anxiety or unease goes away. So like you're doing it just because without doing it, you feel like you feel very bad. Compulsive is worse. And impulsive is when you do it without forethought and without considering the consequences. So you move from impulsive behavior to compulsive, and that is scary. This is not unusual, as every learning or action moves towards automatization to free up your brain's cognitive resources. Initially, when you start driving a car, you have all your attention resources, all your resources towards on the road and driving. But slowly, as time passes, it's easy for you to drive a car and listen to someone talking, because your brain has sort of automatized the activity. And this is what happens in addiction as well, which is a bad thing here, because you aren't learning anything cool. But this is how your brain works. So this is a study related to internet addiction. You can see a picture at the left bottom corner. So the study was related to internet addiction. If you remember, the previous slide was about cocaine addiction, and it was a substance-related addiction. This slide is about internet addiction. So this is a behavioral addiction. The study showed a reduction in gray matter volume in an area almost similar to the substance addiction related area. The yellow region is actually a part of the dorsal anterior cingulate cortex. There it was the right cingulate cortex, if I'm not wrong. So it's a similar area, and this is responsible for decision making and conflict resolution. The primary takeaway is there is something more to addiction than the substance or the behavior that causes it. It's not that cocaine causes addiction, not your phone that's causing addiction, but it's probably a set of responses in your brain to a set of stimuli when presented repeatedly. So, and whether one gets addicted or not, what they are addicted to, like food, cocaine, alcohol, it's all controlled by multiple genetic and environmental factors. And this field still has a lot to discover. Okay. Now, the next two slides will be talking about why can't you just willpower your way through getting rid of addiction? Why is addiction so sticky? Right. There are 3 stages of addiction. Before I go into that, just take a half a minute breather so that we let things settle in a little bit. And we realized what's going on in our brains during addiction. We saw that there is an overlap between substance addictions and behavioral addictions. Now we'll see why addiction is so sticky. Okay. So the first stage of addiction is binge and intoxication. This picture is going to show you how initial use progresses and your behaviors change because of the kind of changes happening in your brain. Why do people react differently towards addiction? For example, tolerance levels are different. Because of multiple factors, because your genetics might be different from mine. So your genetics not only determine what you get addicted to. For example, you might be someone who might be more likely to get addicted to food, maybe junk food, And I might be someone who gets addicted to alcohol. That is one. And whether you get addicted or not, that is also genetically determined. The second factor is your environment. You might have a vulnerability in your genetics that gets emphasized or aggravated due to some environmental factors. For example, you might turn to a certain substance or behavior if you repeatedly have bad social experiences or you don't have a supportive environment. So you can't really say there's this that's responsible where people react differently, but multiple factors. Okay, I hope that answers your question. Right. So I was talking about the first stage of addiction, which is the binge and intoxication stage. Initial feel-good experiences related to the substance or behavior creates associative learning. Associative learning, I hope you know about Pavlov and the dog. Which means just the cue related to or predicting the addictive substance or behavior is enough to cause dopamine release. Like I told you about sight of food making me want to watch my favorite TV series. So this makes you crave for it and you finally go ahead and do it or you binge on it. That's the first binge and addiction stage, binge and intoxication stage. The second stage is of withdrawal and negative affect. As we've talked about earlier, the dopamine flooding leads to a hypo-functioning reward system, which means your reward system can't experience pleasure anymore. You can't experience pleasure anymore. Not only that, our stress and negative emotion reactivity is increased because of changes in the amygdala. The dopamine chemical that was getting released, I told you that it affects different parts of your brain. Amygdala was the part responsible for emotion control. So that guy is also affected. What happens is now you are more sensitive to experiencing stress and negative emotions. Even the littlest thing can cause release of cortisol and you will feel stress. So it's too bad because you aren't using the drug or doing the behavior because it feels good. It initially felt good, but now you're doing it because you want to escape the terrible feelings that are in its absence, that are there in its absence. So that's withdrawal. That's the second stage. Now, the third stage is the preoccupation and anticipation stage. As you can think from the name itself, you're preoccupied with the substance or the behavior. So changes in the prefrontal cortex signaling, the guy responsible for decision making, makes the individual unable to resist the urge even if the intention is in place. There's also continuous thoughts about the drug or behavior that occupy the individual's mind because of hypersensitivity to cues. You see something, you're immediately reminded of that thing. And slowly, everything is a cue. Even a friend you sat with to drink alcohol, maybe, just the sight of the friend will remind you of alcohol. So this is a whole cycle where neuronal pathways change. It's called neuroadaptation. And you can stick around and Erin will be talking about a little bit of that. So what happens is it's your body's way of striving towards homeostasis, which was disturbed by long-term addictive behavior in the first place. This picture shows the behavioral changes where the major highlight is progressive loss of control. Right. These are the last two reasons that you can think of addiction as sticky. The first one, you can see a little kid sitting in front of a marshmallow. I hope you guys are familiar with the Marshmallow Experiment. If not, I'll be talking about it. Dopamine is amazing at highlighting available goals. There's a dopamine surge whenever associated cues pop up. So available goals are only highlighted. And whatever else is important, that is not on your mind because dopamine does the magic and it's gone from your head. It inhibits the awareness of competing goals. This is exactly how the beam of focus narrows down on the present goal. So in this marshmallow experiment, these kids were told that you have a marshmallow in front of you. You have a marshmallow in front of you, and if you wait for 10 more minutes, I'm going to come with another marshmallow and you'll be given 2 so that you can have 2. It was so difficult for these kids to resist the temptation because that thing was right there, and the dopamine in their heads was highlighting that available goal. Even though 2 is better than 1, it was the part of the brain that was responsible for more important decision making that wasn't mature yet, and they had a very hard time resisting the marshmallow. Also, even adults exhibit delay discounting. This is this whole phenomenon is called delay discounting. That means closer goals or available goals are seen to be more attractive than goals that you don't see now. So even adults exhibit this behavior when their dorsolateral prefrontal cortex is artificially confounded using a mechanism called TMS, and they make terrible decisions. Okay, so the second part of the slide is about ego depletion. If you can see that this man is walking to, he can see a bakery every day, maybe 3 times during his day. He keeps resisting the temptation of eating the cake, and by the end of the day, he ends up eating the whole cake. Now, what's going on? It's something called as ego depletion. It's the loss of self-control. I'll address your question by the end of this slide. So ego depletion is a loss of self-control. The more you employ your self-regulation and control resource, the more it gets depleted. So it's like a muscle that gets fatigued after holding a heavy weight, and it'll just collapse after a while. So if I hold a weight in my hand, I can't keep it up the entire time. Similarly, you can't keep resisting a temptation for a long period of time. It's what happens where dieters end up overeating by the end of their restrictive diet. So this is something that is also involved in addiction. Now you guys might be wondering, I was supposed to talk about smartphones and I haven't really gotten to them. This slide is what is going to delve particularly. This slide is going to delve particularly into what happens with regards to smartphones, what makes them so addictive. Okay. Before that, there was a question. If a person has a lot of negative thoughts and stays unhappy on a daily basis, this person is not on any drugs. Could you please explain what happens to this person? So whenever, if I'm understanding your question right, I understand that there is a person who is continuously unhappy and they are not on drugs. How do they deal with it? Usually when people are unhappy, if they have access to drugs and exposure to drugs, then they are more vulnerable and they might get into drugs or different addictions because they are at a higher risk than others. But if they are not doing drugs, they might have other coping mechanisms. For example, they might have healthier coping mechanisms. So even social support is something that is a factor that can save you from a lot of addictions. Okay. Yeah. I'll just end this last slide. Here is where we spill into the area of persuasive design. So we know that there's a lot of fine restructuring and personalization of content that goes on depending on how you use a particular platform. So the algorithms create a content bubble that is bound to get you to click. For example, if you use Instagram and you click on a particular post, Instagram create similar, the algorithm of Instagram makes sure you get similar kind of posts so that you're more likely to click on them again and again. So by catering to your behavior patterns, they are harboring your brain's reward pathway to take over. Now, there's also push notifications that you continuously see popping up on your phone. For example, if you have a mail that will let you know that you have a message from someone on LinkedIn, but it won't tell you what it is. Instead, it'll want you to get onto the website. Pure curiosity and variable ratio reward patterns are harbored to increase app use. You just you don't know when you're going to get the reward, so you keep checking and checking. There's also no stopping cues, which means you keep scrolling. There's no end to the scrolling. It's a clever way for you to not understand when to stop. And they harbor a natural affinity to certain colors. They've conducted experiments which show that we are naturally, our eyes go towards red and warmer colors. That's why they harbor that and design their platforms based on those colors. Finally, we are social animals, and any kind of social contact is quite reinforcing. The addictive properties of smartphones stem from hypersocial environments. So they use variable ratio rewards. You will keep us hooked, and you don't know when you might get a reward. So you keep checking, and it's so convenient for you to just pick it up and check it. These are some factors that make smartphones addictive, and that's the end of my slides. I think we've addressed quite a few questions. If you have more questions, you can mail us at this. All right. Thank you.
Awesome. Okay.
Suman?
Oh,
Maybe I missed something. Could you please tell us about the withdrawal effect? How does it work?
Okay, I'll just tell you quickly in like a sentence. Withdrawal is initially you are doing a substance use because you like it, but withdrawal is when you do it because not doing it makes you feel bad. So when you're not using your phone, you constantly are anxious and you feel very uneasy. So that feeling, to get rid of that, you go back to your phone. That's withdrawal.
There was mentioned you experience anxiety, fatigue, and nausea sometimes during withdrawal. Why is that so?
That is dependent on the drug that is being used. So it's not all behavioral addictions might not show nausea or fatigue really, but depending on what drug is being used, withdrawal symptoms might be different. I think we're running on a time crunch a little bit. Yeah.
All right. Yeah, that was a fantastic presentation. I personally learned a lot about the neuroscience of addiction and why smartphones are more dangerous than we think. So thank you so much, Suman. And so next we'll be moving on to the presentation on the neuroscience of learning, which I will be presenting. And so I have a passion of neuroscience and I aim to do research in the future. I currently have a research, my name is Erin, by the way. I didn't actually say my name. My name's Erin. And I currently have a research position for studying aphasia, which is really cool. And if you have any questions about that, feel free to put them in chat or anything like that. If you're on the SN Slack, feel free to message me there. And then I've been an SN member for about a year now, and I currently serve as co-director of SN's Brain Resource Creation Department, or BRC. So we are on a bit of a time crunch, so I will share my slides and try to go through those quickly. And I hope that I hope you all are okay with going a bit over the ending time, which is technically in about 10 minutes. But if you all can stay, that would be amazing. But it's on your own schedule. This is for you all. So I will share my screen now and we'll get started. All right. Okay. Hopefully the slides are actually working well. Okay, so yes, welcome to the Brain Resource Creation Workshop. This is presentation number 2 on synaptic plasticity and the rate of learning. All right, so first I think it's important to get a little bit into the history of synaptic plasticity before diving into what it actually is. And also I'm going to talk a bit quicker since I do want to respect your all's time. So if I'm talking too quickly, please just let me know in chat. I'm looking at it as I go, so just let me know there. Or anything else, any other questions that you have. All right, so Plato, the ancient Greek philosopher, described the brain as a wax block that may vary in size, cleanliness, and consistency in different individuals. And so this is still considered relatively accurate today, actually. So before the 1950s, researchers actually thought the brain was static and that they remained unchanging after adolescence. But Donald Hebb kind of changed the game and stemmed off of Plato's description of the brain. And Hebb was a psychologist from the early 20th century, and he suggested that neurons that fire together wire together. That's a very, very important quote, so I'll repeat it. Neurons that fire together wire together. And so this means that if 2 neurons fire at the same time, or a collection of neurons more so, then their connection will become stronger, which in essence is synaptic plasticity. So let's see, what did Donald Hebb, the psychologist from the early 20th century, suggest that neurons do? Stemming off of Plato. Oh, also, for addressing the message that I got privately, did you mean my email or Suman's email? Because those would be different. And then if you want Suman's email, that would be, oh, mine. Okay. I will type that to you. It'll also be shown at the end of the presentation. I'll go ahead and send that to you. All right. So we do have an answer in chat. Neurons that fire together, wire together. Fire together. Yes, yes. That is awesome. That is correct. Congratulations. So yeah, he suggested that neurons that fire together, wire together, meaning that a collection of neurons that are activated together grow stronger as a unit, and that can translate to a variety of different things, such as learning a new skill, which we'll get into a bit later. All right, so now we're going to talk a little bit about the anatomy of neurons. So essentially, you have dendrites, which receive the incoming signal from the postsynaptic terminal of the previous neuron, and dendrites have so the presynaptic neuron terminals are on the sending cell of the previous cell, and then there's a synapse, which we'll cover in the next slide, and that comes into the postsynaptic terminal on the new neuron, and that is received by the dendrites. And the nucleus, which is also called the soma, it's the cell body. And so the soma contains the nucleus, and the soma is the cell body of the neuron. And then the axon is what transmits the electrical signal called the action potential. And then there are also Schwann cells, nodes of Ranvier, which I won't go too much into it, but essentially, if you don't already know how axons or how the action potential moves across the axon, is helped through myelination, which it mostly consists of fats, and it insulates the axon essentially to be able to transmit the action potential or the electrical signal faster. And so myelination results in faster, more efficient transmission of messages throughout the brain. So it's super, super important. There's a variety of diseases that are associated with a lack of myelination. So myelination is super important to transmit messages efficiently. And nodes of Ranvier, so essentially the action potential or the electrical signal doesn't exactly jump, but it's not a linear passageway. It is more of a jumping motion, like a sine graph. Have you ever seen that? It's kind of like that across the nodes of Ranvier. So those are the breakages in the myelin. And then the electrical signal goes all the way down the axon to the axon terminal, which contains the presynaptic terminals, which in essence is just the synaptic terminals that contain vesicles, and they send off the neurotransmitters of the electrical signal, things like that. So I hope that was clear. Do y'all have any questions on that? I can explain anything again if need be. Okay. So just to recap really quick, so dendrites receive the signal and it goes through the myelinated axon to the axon terminal where it gets sent off to other neurons. All right. So an intro to synapses. So this will be between the presynaptic and the postsynaptic terminals, where the electrical signal gets transferred through via chemicals called neurotransmitters. There are electrical synapses as well, but it's most commonly transferred through neurotransmitters, so chemically. And so chemical communication is more common than electrical communication. And so that's why we're focusing on that this slide. And so the steps for chemical neuronal communication are the first, so the action potential from the neuron's axon reaches the synaptic terminals. So I never really explained this, so I'll go over what is actually an action potential. So in brief, it's an electrical signal that travels on a neuron's axon that is hopefully myelinated and activates the neuron to send a signal to another neuron. And so the action potential causes ion channels to open and close, and this causes the chemical neurotransmitters to be released into the terminal, and vesicles, which are little tiny bubbles of sorts, connect to the terminal and are released into the synapse. And so neurotransmitters are chemicals that bind to the receiving neuron's receptors in order to make it more or less likely to fire. This is essentially communication throughout the brain. So it's super, super important. And some examples of neurotransmitters are some you might have already heard of, including dopamine, like we talked about in the last presentations. That has a lot to do with happiness, addiction, different things like that. Even some memory formation through the hippocampus. And oxytocin, some maybe that you haven't heard of, like GABA or glutamate. So the excitatory inhibitory, which I won't get into in this presentation, but feel free to look that up. It's really interesting. And then, so once the neurotransmitters have left the terminal, they're in the synaptic cleft or the synapse, essentially. They're different, but we can use them interchangeably for this presentation's purposes. So the synaptic cleft or the synapse. And then neurotransmitters bind to receptors on the receiving neuron's dendrites, like I talked about earlier, and the message the neurons are communicating is made either stronger or weaker. And this ties back into long-term potentiation, which Suman talked about in her presentation. So we do have a question in chat. So how do synapses grow and make connections? What actually happens during LTP at a cellular level? So synapses grow and make connections through, it happens in different stages. So when you're younger, there's something called synaptic pruning that happens that clears out all the neurons that you aren't using as often. So referring back to the quote, neurons that fire together, wire together. So the neurons that aren't firing or wiring get pruned out, and that's essentially development of the human brain and animal brains in general. And so yeah, so if they're firing together, they wire together. And what actually happens during LTP at a cellular level, I'm not terribly familiar with that. Maybe Suman can help answer in chat. But essentially long-term potentiation, if the neurotransmitters are getting sent, so if a neuron is being used very often, the myelinated axon is getting traveled on very often. If that's being used, then the receptors on the receiving neuron cells will become more receptive to the neurotransmitters being sent by various neurons that it connects to. So that's essentially what it is at the cellular level. I hope that's not too broad. I hope that answers your question, but and then feel free to put any further questions in chat if you want something explained better. All right, so now what chemicals are released into the synaptic cleft or the synapse and bind to other neurons? And if you can give some examples, that would show a high level of learning. What do you all think? I can give you a hint. It starts with an N. What do we think? I can repeat the question. So what chemicals are released into the synaptic cleft and bind to other neurons? Neurotransmitters like dopamine, oxytocin, GABA. Awesome. Yes, that is correct. All right. So let's get into the meat of this presentation. So what is synaptic plasticity? So now we have a good handle on neurons and their synapses. So now we can get into the communication of those neurons and how essentially that leads to changing how your brain is wired. And so synaptic plasticity is a process by which the synapses between neurons change structurally and or functionally. So it also controls how effectively two neurons communicate, meaning it can change the pathways of neuronal communication to be stronger or weaker, which is essentially long-term potentiation. There's also LTD, which I believe stands for long-term deterioration. There's another one. There's another one that is the opposite of long-term potentiation. I need to dust up on my LTP and LTD. But so on the next slide, we'll cover why synaptic plasticity is important. And you can kind of see in this image as well to help explain the question that was asked. So the neurotransmitters are being sent through the presynaptic terminal to the receptors on the postsynaptic cell through the synapse. And so those ion channels are being activated, those neurotransmitters are being sent. And so those neurotransmitters or the receptors can also grow in number. So that's another thing with LTP is that they can not only become more receptive to the neurotransmitters, but they can grow in number. So that's another way that connections between neurons can become stronger or weaker. So the opposite would be, less receptors on the postsynaptic cell, or they become less receptive, and so that would result in a weakening of the connections between neurons. And that can happen as you get older with Alzheimer's and things like that, and also synaptic pruning when you're younger. All right, so why is this important? So synaptic plasticity describes the ability for our brain to change throughout our lifetime, and that's the basis of learning, memory, adaptation, all of these important metacognitive things that are essential to life. And so one example is, I'm sorry to bring this up also, but do you feel like a new person during the pandemic? So that actually results from you adapting to the new situation. And the situation has unfortunately been lasting a very long time, but that is essentially what that is. So if you feel like you've adapted more so when we were doing online learning, I hope not too many of you are still doing online learning, but when you were, you probably adapted to the situation and were able to learn somewhat in that situation. That would be the result of synaptic plasticity. And so another example is that is in taxi drivers. And so studies have shown that their hippocampi, which is again the structure in the brain that partially deals with spatial navigation, memory formation, like Suman mentioned, has a lot of different functions. And the hippocampi are actually larger in taxi drivers. And so this is because synaptic plasticity is involved with a wide range of things, including, okay, that was actually going on. So taxi drivers, those receptors are growing in number. So it's the spatial navigation. So they have to know by the back of their hand, they have to know very, very well the geography of a city, its streets, various things. And so they need to have all this information in mind. And so if the hippocampi is dealing with spatial navigation, those neurons within the hippocampi are going to be used way more than than someone who's not a taxi driver. And so there's going to be long-term potentiation within those neurons, and they're going to have stronger connections. And so synaptic plasticity is involved with a wide range of things, including forming our earliest attachments to habit-forming behavior throughout our lifetimes. And so as another example, chewing on your nails, maybe it helps you release stress. This means your neurons that are connected to nail-biting pathways will be increasingly activated when you're stressed. And so this also kind of connects back to the conditioning and what Simone was talking about, how you can become kind of more attached to different things and the habit formation. So if you become stressed, if that cortisol is released, you might become more likely to chew on your nails and do, have that. If that has released your stress in the past, reduced your cortisol level, you're going to be more likely when cortisol is released to chew on your nails. All right, so how does this work? So the main feature, and this will be an important wink, wink in an upcoming slide, is the gradual increase or decrease in postsynaptic receptors. And so these are the structures that receive neurotransmitters and that trigger an electrical signal, which again is the action potential. And so as you can see in the picture, neurons adapt to sustained stimulation, meaning they can alter the amount or type of receptors they have. So certain neurotransmitters will be more likely to bind. So that changing the type of receptors they have can also be a part of growing more receptive to the neurotransmitters. That helps with long-term potentiation and strengthening the connections between the neurons. Right, so let's test what you've learned so far. No pressure though. What is the main feature of synaptic plasticity? Maybe I can sneak back to the other slide real quick. Never mind. Can I go back? Yes. So what is the main feature? What do you all think? Okay, just for time's sake, and it is a kind of a long answer, so I understand if it takes a second typing it out. Go ahead and keep typing if you'd like, but I'll go ahead and say it. So as seen in the last slide, it's the main feature is the gradual, the gradual increase or decrease in postsynaptic receptors. So yep, you got it right, affecting postsynaptic receptors. Awesome. All right. So now let's actually, long-term depression, that is it. Yes, so LTP and LTD. So kind of expanding on these concepts. So one of the ways that LTD, so long-term depression, is involved in memory and learning is that it's thought to help in the removal of old and useless memories and skills, like decreasing drug use urges and rehabilitation stages, or unlearning the wrong way to do something. So if you're maybe snapping with the finger that doesn't produce the most resonant sound, your brain's going to change that to, oh, maybe I should use this other. And then you're going to know automatically, okay, I should use my middle finger versus, let's say, your ring finger, which does not produce as that snapping sound that you might want, as an example. And another example is doing something like swimming. So if you're swimming the wrong way and you're like close to drowning every time you jump into a pool, that's not great for your survival. And so if you're swimming the wrong way, you're just kind of thrashing around close to drowning. So LTD would help improve that procedural memory from your mind. And procedural memory is like kind of how to do things, actions, like riding a bike, things like that. And LTP, which is long-term potentiation, which we've talked about, would help you retain the newly learned and presumably correct way to swim, ride a bike, pretty much anything like that, also in terms of learning. All right. And so, like I said earlier, if you have any questions, feel free to put them in chat and I'll answer them at the end of the slides and also at the end of the presentation. So as an example, rodent studies have shown that synaptic plasticity and the nucleus accumbens is central in long-lasting cravings and relapse in rodents addicted to meth, which is a very, very harmful drug. And so why might this be? Well, the nucleus accumbens is often labeled as the pleasure center of the brain. So it's very, very, very much associated with pleasure and dopamine, things like that. And it's located in the basal forebrain. And so you can kind of see a map of the brain. So I don't know if you can see my mouse, but I'm circling over the pink structure above the darker pink structure, which that is the limbic system. So that includes the hippocampi, the amygdala, stuff like that, the thalamus, hypothalamus. And so those are the things that are going to be very involved in memory and learning and emotion. And so that little red dot that you see there is a nucleus accumbens. That's the pleasure center, very associated with dopamine. And it is located in the basal forebrain. All right, so the rate of learning. So this is associated with STDP, which stands for spike timing dependent plasticity. This is central in memory formation and neural networks, so that LTP, LTD. And so the strength of the synapse is, like I said before, strengthened or weakened depending on the timing of the firing of the pre and post synapses, along with the other factors that we've already talked about. So I'll give an example of this to better explain. So the timing of the firing matters quite a lot. And so there's a study that measured the time difference in the pre and post synaptic synapses firing. And the image looks complicated, but hopefully after I explain a little bit, it'll be more clear. So one study conducted measurements that if the timing of the firing of the pre and post synapses in two neurons was less than 0 milliseconds, which they found this by subtracting the firing time of the post synapse by the presynapse. So if the difference was less than 0 milliseconds, this tended to be long-term depression or LTD, or the weakening of the synaptic connections. And if the difference of the firing time of the post minus the presynaptic firing was above 0 milliseconds, this was shown to lead to LTP, long-term potentiation, or the strengthening of synapses. And so you can see all that info visually on this image. Yeah, so you can see the so before the firing, before the postsynaptic neuron, which, and if it was below 0 milliseconds, then that led to the LTD, and then if it's above, that led to the LTP. Hopefully that makes sense. It is a bit of a complicated study. So we do have a question. Any future research on meditation related to addiction and changes to increase on myelin and decrease in addiction? So meditation is very interesting. I want to look more into it. So I do believe that meditation has been linked to LTP. I would definitely recommend researching it a bit on your own. You can go to a variety of different databases like JSTOR, PubMed. If you're not familiar with those, just type in those. I can PubMed, JSTOR, and there's a variety of other databases. Oh, and Suman has me covered as well. So she said, regarding meditation, there's been research which says that meditation changes cortical volume. And hopefully that answers your question. But yeah, I would definitely recommend going into these research databases. There's dozens upon dozens of research papers on so many topics, and I hope including on meditation related to addiction. You can just type in keywords into the search bar within those databases, and you'll find a variety of different papers, and you can kind of read through those. All right. So now let's get into an example. So this was a study conducted by Dr. Saki Louie at Wesleyan University, and she's a neuroscientist at the university, and she's also an accomplished violinist. And so she found that the right hemisphere motor cortex, which controls the left hand, is used for precision by violinists and has more structural connectivity in those who play the instrument. And I would believe this goes for other instruments as well, like flute or saxophone. And so the younger the violinist starts playing, the greater the structural connectivity, because this relates to the synaptic pruning. So the younger you are, the more your brain is pruning, taking out those neuronal networks and neurons that don't fire as much, and strengthening those neurons that do fire, and the groups of neurons, which, to kind of make this concrete, ties back to LTP. And so if you're playing an instrument, not just a violin, any instrument at a younger age, those neuronal connections are going to be strengthened because they're being used, especially if you practice very often. That's also important. If you're working on a skill, if you're practicing every day or even every other day, those neuronal connections are going to be strengthened because you're practicing. And if they're not being used, then they're going to be pruned out because you're not using them. So what's the point of having them in the brain at all? And so this increased structural connectivity is caused by synaptic plasticity, and mechanisms like LTP allow for better communication between neurons. So that skill, the more you practice, and especially the younger you start playing, you're going to have those neurons strengthened in accordance to that skill. So that was a very cool study. All right, and so here are some resources to learn more. There's the edX Fundamentals of Neuroscience courses. It's a 3-part course, and it's a fantastic introduction to neuroscience. And there's the Khan Academy nervous system videos. I will send a link to this presentation in the chat after the presentation. And so you all can go back and click on these links and explore them a bit. All right, so I really hope you enjoyed the workshop. And do you all have any questions? Is there anything that you're still a bit confused on? I can go back and we can explore that slide a little bit more. We can talk about anything, discuss any questions you all have. I'll stop presenting now. Can't get off the slide. There we go. Okay. Do you have any questions? Thank you. Thank you. Well, if you don't have questions, then that concludes our BRC workshop hour for today. Thank you for sticking around. For those of you that did, we did go a bit over time, but that was because we had so much amazing stuff to say about our brain. It's an incredibly complex but fascinating organ. But yeah, so we hope you enjoyed the session, and I will get a link to my presentation. Suman, if you're comfortable with sharing your presentation, feel free to put that link in chat. I would recommend setting it on they can view, so the presentation isn't altered anyway. We trust you all, but just in case.
I'll just put it up.
Okay. So here's a link to my presentation. I hope you all are able to access it. You should be able to. But yeah, so, and just so you know, we plan to host these meetings, these BRC workshop hours each month with new workshops each time. This is our second one ever. Last month was the neuroscience of learning or of dreams and memory. That was super interesting. That is on Simply Neuroscience YouTube. So that's up and that is free for your viewing if you're interested. This one was on the neuroscience of addiction and the neuroscience of learning. And so in order to be in the loop about future workshops, stay tuned to the Simply Neuroscience Instagram for announcements regarding next month's session. So if you have any other questions, feel free to email us at our SN emails shown on the slide. So that is not being presented right now, so I can put it in chat. And Simone just shared her presentation, so feel free to explore that. And then someone asked if we can conduct another workshop on psychiatric disorders. I do believe someone within the BRC department is working on psychiatric disorders. I'm not sure if that is you, but if not, we do have that presentation in the works, so stay tuned. I can't guarantee that it'll be next month, but that is in the works, so that is super exciting. So I'll put my email in chat.org. I also put my, so that is my Simply Neuro email. And then Suman, feel free to put yours in chat as well if you're comfortable with that. All right. I will stop the recording now, but thank you all so much for attending, and I hope you all have an amazing day.